Good evening, everyone, and welcome to the Brian Crombie Radio Hour on Saga 960. As any of you who follow my show know, I like to expose you to interesting technologies and companies, particularly in the life sciences area. And a gentleman that I think very highly of, John Ruffolo, previously recently promoted a company because it's very helpful to him and some of his challenges. And it's a nerve regeneration company. We're going to hear all about it. It's called NervGen. And today on the show, we have Dr. Harold Panett, co-founder of NervGen Pharma, Mike Kelly, the president and chief executive officer, and Dr. Jerry Silver, a co-inventor and the scientific advisor for NervGen Pharma. Maybe we could start with you, sir, Dr. Harold Panett, and tell us a little bit about what NervGen is, what the technology is, and what you're trying to solve.
Well, I think that's probably better. I'm going to hand that over right away to Mike because he's the CEO and he can give an overview. As the founder, I can give you an idea of how the company started, but I'm going to throw that one back to you, Mike.
Okay, sir.
Sure.
Mike Kelly, the president and chief executive officer. What's NervGen all about, sir?
Sure. NervGen is it's a small company. We're based in Vancouver, Canada. We have operations. We're a virtual company, so we have operations in Toronto. I'm outside of Philadelphia, and we have several employees throughout the United States. But we're a development company. We have an interesting product candidate that just entered phase 1b/2a in spinal cord injury. It's based on technology that we'll talk about today that was discovered in Dr. Jerry Silver's lab. But we're a small company that has the ability to show that the body can actually repair itself when patients have either experience an injury like a spinal cord injury, or the other diseases we're looking at, or the other afflictions we're looking at are stroke, multiple sclerosis, Alzheimer's disease, ALS. So whenever you have damage to a nerve or central nervous system, the body actually, is very good at protecting itself, but it's not very good at unprotecting itself and allowing nerves to regenerate. So I'll let Jerry get into the specifics of his discovery there. But
this is NervGen Pharma. So is this a pharmaceutical product, I presume, that you're using?
It is. This is a pharmaceutical product.
So there's a company here in Toronto that uses electrical stimulation. So I've heard of other technologies that use other modalities to repair nerves. This is the first pharmaceutical product I've heard. Are there other pharmaceutical products on the marketplace to address nerve damage?
No, there's currently nothing on the market to work on nerve damage. There's products that are in development, earlier stage development. Several larger companies are working in the same space as well. AbbVie has a program, Mitsubishi Tanabe has a program, but nobody is as far along as we are in phase 1b/2a human trials. We started our study in chronic patients that have a spinal cord injury. We're expecting to enroll a group of patients. When I say chronic, they've had injury between 1 and 10 years ago. We're expecting to complete enrollment in that group the end of this year, the first part of next year, and we'll have results mid-year next year. So it's a very exciting time for the company.
Fantastic. Well, I look forward to hearing more about it. My son broke his arm and quite severely, and lost all ability to move his hand and his wrist. But they said it would grow back. The nerve would grow back an inch a month, and 6-7 months later, he regained the ability to move his hand, and that happened naturally. So I guess I'm a believer that the body can heal itself, but that's just an arm, not a spine. So I look forward to hearing what your solution is. So, Dr. Jerry Silver, over to you, sir. How did this all come about?
Did you want to let me address this question or Harold's?
Why don't we come to you, and then we'll go back to Dr. Plunkett and hear about how we found this all about.
Yeah, I'll let Harold start, and then I can.
Okay. Sure. So a bit about my background, Brian. I'm a recently retired dentist. I was a dentist for just about 38 years in the community just outside of Vancouver, BC. I had no inkling, desire, or suggestion that I would found a biotech company. But about 7.5 years ago, my wife and I were on holiday. I got a phone call late one night from my son, and he said, "Dad, Cody has fallen." That's his wife, Cody. And she's fallen and she's broken her back. And then I was trying to assimilate that information. It was just so shocking. And then he said, "And now she can't feel her legs." So we rushed home the next day. We arrived to the sad news that she would never walk again. She was what's called a complete T11 paraplegic, which means she has no feeling, no sensation, no motor, no movement below the level of her injury, which is above the level of her belly button. Pretty devastating time for her. She was 29 years old at the time, 3 young children. Anyone listening to this who has children will understand the concern and impact that this had. Her children were only 18 months and 4 and 6 years of age at the time. So basically all our lives were sort of, thrown up in the air. We had no idea what the future was really going to hold for her. And at that time, I just decided I was going to try and do something about it. I wasn't really clear what I was going to try and do, but I had enough medical background that I could read the literature. So I sat in front of the computer. I went onto the search engines and I started to read every paper I could find on spinal cord injury. When I found something interesting, I would contact the researchers, and I ended up talking to and emailing researchers all over the world, all over Europe. One in Russia, a couple in Israel, all over North America. And I really wasn't finding anything that I felt I could really get excited about. There's a lot of people doing a lot of good work, but nothing really that I thought was foundational or offered a revolutionary approach to helping Cody. Until I found Jerry, and I read his 2015 Nature paper, and I got excited. It was completely different from anything I'd read. I thought he'd found something foundational. I thought it was very special. And so I got a hold of him and I phoned him. And I said, "Jerry, I'm a dentist. This has happened to my daughter-in-law. I like your technology. I'd like to form a company, raise money, and turn it into a drug." And he was very kind and very polite. And we spent that summer of 2016 talking. I don't know if it was every day, Jerry, but I think between emails and phone conversations, it was pretty much every day for 4 months. And by the end of that 4 months, I was more convinced than ever that this was something that was pretty spectacular. So I didn't know until 2 or 3 years later. Jerry told me over dinner one day that he got off after our first phone call. He hung up the phone, he turned to his wife and said, "Well, that guy's crazy. That'll never happen." But here we are. It's maybe I was crazy, but it's happened. And as Mike said, we're in our first, we're in our proof of concept phase 1b/2a trial, and we're injecting spinal cord injured patients. So it's been quite a ride. It's been almost 8 years, but here we are, and it's, as Mike said, just phenomenally exciting time for the company.
Phase 1, phase 2, Dr. Silver, is a few people. How many patients are you dosing right now?
I think it's 20 patients, 10 spinal cord injured and 10 placebo.
Excellent. And we'll delve into a greater detail, but assuming you're successful, you then go into a full phase 2 and then a full phase 3, or what's the strategy from here?
Only Mike can answer that one.
Yeah, sure. So the goal here is there's actually 40 patients total in the study. There's 2 different cohorts. There's a chronic cohort that has been injured between 1 and 10 years ago. So there's 20 patients there, 10 drug, 10 placebo. And then there's 20 patients that are in what's called a subacute cohort, meaning their injuries occurred between 10 and 49 days ago, 10 drug, 10 placebo. So the goal is to learn from this study if we can translate what Dr. Silver has proven in preclinical models and animal models, if that translates into human model, and if it does in any way, shape, or form close to what it did in animals, then we're off to the races. And we think that we'll do at least one other additional study, and we're going to sit down with the FDA and get a path forward from there. And
would you get an orphan drug designation that would mean you can go more quickly to commercialization, or would this have to go through the typical, 500 people in a phase 3 massive study?
No, that's a great question. And we actually just announced last week that we received fast track designation from the FDA. So there's several different pathways that the FDA has laid out for companies like ours. Fast Track is one of them, is the first one that we've applied for, and we've received it, which basically means that, number one, there is nothing for these patients, and it's a devastating unmet medical need in the marketplace. And number two, your data that you've delivered to the FDA has indicated that your product has significant hope and promise. So we checked those two boxes. The FDA gave us an announcement last week that we announced to the world that we received Fast Track. After this clinical trial, we'll be filing for breakthrough, depending on obviously the clinical data that comes out. But breakthrough designation is Fast Track on steroids. It gives you a little bit more personal relationship with the FDA. All of these designations we'll be filing for over time as we progress the product towards the market to expedite this and get this to patients as quickly as humanly possible.
Sounds very encouraging. We're going to have to take a break, though, for some messages, and we're going to be back in 2 minutes with the team from this company, NervGen Pharma, talking a little bit more about what this product is, how it works, how we can actually repair our nerve system, and what the prospects are for this company and for the technology and for the patients, most importantly, that suffer from these spinal cord injuries. Stay with us, everyone. We'll be back in just 2 minutes. Welcome back, everyone, to the Brian Crombie Radio Hour on Saga 960. Tonight, we're talking about spinal cord injury, nerve damage, and a really interesting Canadian company, NervGen Pharma, and three executives from that company, and about what their pharmaceutical product does, the clinical trials that they're involved in, and what the prognosis is for both this company as well as the technology, and most importantly for the patients involved. Our guest tonight are Dr. Harold Panett, co-founder of NervGen Pharma, Mike Kelly, the president and chief executive officer, and Dr. Jerry Silver, the co-inventor and the scientific advisor to NervGen Pharma. As I mentioned, NervGen is on the TSX Venture as NGEN, a clinical stage biotech company, steering Canadian biopharma to its first human clinical trials as the company proceeds with its landmark clinical trial for NVG 291 for chronic and subacute spinal cord injury patients. Excellent. So maybe I could ask a couple of questions, and I'm not sure who wants to jump in, but you guys can just decide. Let's talk about, to start with, how big this problem is. Mr. Kelly, do you have a sense of how many people actually have spinal cord injuries in Canada, or the United States, or North America? How prevalent is it? How many people are suffering from these incredible life-changing problems?
Yes, absolutely. There's roughly between 18,000 and 19,000 new cases each year in the United States. There's a pool of 300,000 people in the United States that are living with spinal cord injury every single day. I was at a conference where I heard a Canadian health ministry person speaking, and said that it was the largest financial impact to the health industry in Canada. It's a massive impact to the U.S. health industry. These patients, the cost per patient in publications ranges between $1 and $4 million per patient over the course of their lifetime. It's a $54 billion U.S. problem. That and it's not only a financial problem, as Harold originally mentioned. There's a lot that these folks go through and live through, emotionally, physically, and financially. So this is a massive problem that needs to be met head on.
It sure sounds like it must be. Is there currently a standard of care? Like what does one do now? Is there anything to do now?
No, there's typically the standard of care is the patient has an injury, and they typically will be hospitalized. In the first 2 months, they'll have surgery, and then they have a very rigorous rehabilitation process. They push themselves to push their bodies to get as far as they possibly can, and then they typically improve over time. But typically somewhere between the 9 and 12 month mark, it starts to, simmer down as far as the gains that they've made to improve, whether it's hand motion, leg motion, walking. And a lot of it depends on how severe their injury is, how young they are, how physically fit they are, and honestly, how hard they push themselves through that rehabilitation process. But they will plateau in the 9 to 12 month time frame. And there's nothing out there for them other than, right now there's a lot of drugs in development. There's a lot of stimulation protocols in place to see if they can connect the plasticity of teaching the body how to regain function over time. But nothing is on the market today.
Maybe I could turn to Dr. Jerry Silver and ask you, sir. I understand that you are professor of neurosciences at Case Western Reserve University in Cleveland, Ohio, and you've, I've been told, have overturned nearly a century of neuroscience precedents based on your understanding of CNS injury. Can you tell me, what you did and how you found this new pharmaceutical product?
Yeah, well, I'll make a 40-year journey very quick, if I can. I started out as a developmental neuroscientist. I got my PhD in neuroanatomy. And at the time, this is the end of the 1970s, early 1980s, there was a large interest in the developmental neuroscience world about the factors, the cells and the molecules that guide the nerve cells to where they ought to go during development. And people were looking for signals, molecules that told the nerve cells where to go. For instance, from the eye to the brain, or across the midline of the nervous system. Why do axons grow where they do? And I became interested in that problem, but I asked a different question, one that at the time nobody was asking. And I asked the question, why nerves don't grow where they don't? Very often in the developing brain, the nerve cells, as they're growing, turn at sharp borders, or they refuse to cross certain planes from one side of the nervous system to another. And I always wondered if there was some kind of a guardrail that might exist, a boundary or a barrier at those points of decision-making. And I started looking at those positions where nerves turn. And there are a number of different regions. One I focused on in the spinal cord. At the very top of the spinal cord are a group of cells, they're called glia. And at the very top of the spinal cord, those glial cells, those non-nerve cells, keep the nerve fibers on one side of the spinal cord or the other. And they never cross that region of glial cell existence. And I always wondered, what might those glial cells be making that might form a boundary or a barrier to crossing of nerve fibers in that region? So we looked very, very hard. It took many, many years of searching for a molecule that was known to be inhibitory long before I got to study them, but might be in those glial cells at that guardrail. And in the mid-1980s, we discovered that a family of molecules called proteoglycans, they're called proteoglycans because there's a protein, proteo, to which are attached long-chain sugar molecules called glycosaminoglycans. So they're called proteoglycans. And many of your listeners and you yourself may have seen these products on the shelves of many, many types of companies like Costco, any drugstore.
Proteoglycols or what readily available?
Proteoglycans. And you'll see them in bottles called cosamine or glucosamine or chondroitin or Dr. Smith's joint ointment, a whole variety of pills, cosamine, glucosamine, chondroitin. And people take these molecules.
I take glucosamine every day, so I didn't even know I was
see, so you're quite familiar with it. You're probably taking it because you think that it's improving your joints.
Yeah.
And it
Is
turns
it?
that's probably not true. There have been
Oh.
clinical trials. You may be wasting your money
That
putting
won't be the first
one
time or the last.
You're putting it in one opening and then it's coming out the other, but not doing much for your joints. But people swear by it. Those proteoglycans are extremely inhibitory to growth of cells as well as nerves. They are present in high abundance in cartilage. Cartilage is not innervated. There's no blood supply, and there's no nervous supply. And the reason is proteoglycans fill cartilage. You don't want a nerve supply or a blood supply in your cartilage because it's so bendable. So you can crinkle up your ears and wiggle your nose. It doesn't hurt because there are no nerves in the cartilage. And that's because of proteoglycans. They're very inhibitory. They may be put there in cartilage to do exactly that, to keep the innervation out. Also, they bind a lot of water. And that makes cartilage very, very slippery. And so very slippery, very inhibitory. Proteoglycans used to be called mucopolysaccharides, which is basically cell mucus. And mucus is a proteoglycan-rich product that's produced by many kinds of cells. Why? To prevent a barrier so bacteria can't invade that region or viruses. So mucopolysaccharides with a new name, proteoglycans, are present in abundance in cartilage.
So you found this barrier, but how does this barrier
However,
create a drug?
at the time, it was thought that molecules like proteoglycans were not present, nor could they ever exist in the central nervous system, especially during development or in the adult. But using special kinds of stains and antibodies, we found that those glial cells at the top of the spinal cord that separate the nerves, that don't allow them to cross that border, were filled with proteoglycans. And we told the world that that's what we had discovered. So this is around the mid to the end of the 1980s. And nobody believed that that could be true, that there would be these barrier-like cartilaginous molecules in the central nervous system. But I persisted. I'm stubborn, I guess, and continued upon that challenge to convince people that I was right and continue on with my studies of normal development. But because nobody believed what I was talking about, I actually lost my funding to study neurodevelopment. And then, almost out of desperation, I wondered if after injury of the central nervous system, for instance, after a spinal cord injury, the barrier proteoglycans might reappear at the site of injury or elsewhere and block regeneration. And by God, that's exactly what we found. So we injured the spinal cord surgically. We used the same markers that we did to study proteoglycans in the developing nervous system and found them again in the wound area of the spinal cord injury.
Really? So the nerve—
That's how it all got started.
The nerve isn't broken, it's just blocked by
So what happens after any kind of injury to a nerve fiber tract in the brain or spinal cord is that the cut nerve fibers actually live. But they get stuck and trapped at the entrance to the wound, which we call the scar. Those nerve tips become entrapped for the rest of your life in that region. And so the nerve fibers don't actually die. They just become stuck and trapped. They can't move, as a fly can't move on flypaper. And we found that the proteoglycans in the lesion entrap the nerve fibers. So they're all there waiting to regenerate if possible, but entrapped within the scar. So we figured out a way to break that bond, to release the nerve fibers from that entrapment by finding a receptor on the nerve fibers that stick to the proteoglycans. That was called the so-called sigma receptor. That was around early 2000s. Many, many years passed before we found the receptor. And then we developed a peptide a small molecule that can be given systemically that blocks that receptor. So now the nerves don't even see the proteoglycans. They don't see the flypaper, and they keep on growing. They don't stop. They don't get stuck. And that allows them to regenerate or sprout, get past the point of blockade, and go and grow towards their targets, which they can then.
So this is interesting. You're not actually taking away the blockade of this protoglycade. You're blocking the nerve's ability to sense it and see it.
That's right.
And therefore, the protoglycade's ability to stop the nerve from growing. And
right. You can think of this kind of symbiosis between the nerve fibers and the proteoglycans like Velcro. You can think of the receptor as the hook and the proteoglycans as the loop in hook and loop or Velcro. And what we're doing basically is blocking the ability for the hook and the loop to bind each other. So now instead of hooking up like Velcro does and making an attachment, you just slide past the loop.
And you said you're administering a peptide, a small molecule, orally via a capsule or through an injection or what?
Eventually, it would be wonderful if we could come up with an oral delivery method for introducing the peptide. But right now, it's engineered to get into the spinal cord and or brain via systemic delivery through an injection needle.
And so do you inject it right at the site, or do you inject it anywhere in the body?
It can be injected subcutaneously just about anywhere. And I know that NervGen is using abdominal skin, subcutaneous injections near the abdomen to allow the peptide to gain entrance. It basically gains entrance to the entire body and brain and spinal cord.
And when it goes to other parts of the body, does it do any harm?
Well, we haven't seen any harm yet. No sign of it. It turns out that receptor, the hook that I was talking about, actually increases in those entrapped nerve fibers that get stuck in the scar. So there, the receptor is extremely high. And so the peptide tends to work on those cells first, which is a good thing. But we have not seen any ill effects or untoward side effects, at least in our animal model. So far at all.
You may not know the answer to this question, and maybe I'm just asking you to hypothesize, but why would the body have created this mechanism such that you've got this blockage? Is it because you don't want the nerves to feel pain or something like that, or what?
That's the $20 billion question. Used to be the $1 million question, but with inflation, it's gotten a lot higher. So the reason that this happens is not known. There are species that after spinal cord injury and or brain injury or losing an entire eye can regenerate completely. So goldfish, for instance, if you pull the eye out and cut the optic nerve, it can regrow an eye and regenerate the nerve and can see again. Or if you cut the spinal cord of a fish, like a zebrafish or a goldfish, they'll sink down to the bottom of the pond, feed locally, and then regenerate their spinal cord. And there's even a mammal called the spiny mouse that lives in Australia, whose spinal cord, after becoming injured, cut, can regenerate because it does not produce the proteoglycans. They can regenerate. The spiny mouse can regenerate just about every part of its body.
So does a goldfish not have a proteoglycine?
That's exactly that's right. They do not scar after injury. They build a blastema. They have stem cells that regenerate new nerve cells. They do not produce a scar. They make bridges, and they allow the nerves to grow. And so they have not evolved a mechanism to block growth. Why all warm-blooded species, except the spiny mouse and very immature mammals. So you and I could have regenerated our spinal cords in the womb, in the uterus, about till mid-gestation. Opossums can regenerate their spinal cord in the pouch because they're born so prematurely. But after a certain period, postnatally, for instance, postnatal day 7, if you're a mouse, intrauterine if you're a human, all regeneration stops. And the reason why that happens is absolutely not known. It's thought perhaps that what happens in warm-blooded species that scar is somehow during evolution figuring out that regeneration was not especially productive. And then its job is to wall off and protect the rest of the brain or spinal cord from damage due to all the inflammation. So what the warm-blooded species tend to do is to wall off the area of injury to save the rest of the nervous system from inflammatory chaos. That's the reason it's thought we build scars. But overcoming them or getting rid of them does not cause ill effects. So the answer to your question is we just don't know. It almost makes no sense, because we get better if we allow nerves to regenerate past the scar or sprout, not worse.
Absolutely fascinating. Last question before we take a break, if I could. So why did my son who broke an arm, was he able to actually grow back the nerve?
Very good question. It turns out that the peripheral nervous system over millions of years, because it's damaged so often, especially in our extremities, has developed a way of regenerating itself. It does not produce scar. And the glial cells in the peripheral nerve are different from those that form the scar in the central nervous system. The cells in the peripheral nervous system are called cells of Schwann, or Schwann cells, named after Dr. Schwann, who discovered them. Those cells make beautiful tunnels and bridges at the site of the crush that occurred after your son's broken arm. And those bridges allow for regeneration at about 1 millimeter per day. And eventually, if the nerve is crushed and the tunnels made by Schwann cells are still intact, the peripheral nervous system can regenerate quite well.
God, this is going to go almost political. We need more bridges, less walls.
That's right, absolutely right.
We're going to take a break for some messages and be back in just 2 minutes with this fascinating technology and company that is trying to solve spinal cord injuries and nerve damage. Stay with us, everyone. Back in 2 minutes. Welcome back, everyone, to the Brian Crombie Radio Hour on Saga 960. I've got three gentlemen with me tonight from a really fascinating company called NervGen Pharma. It's a TX Toronto Stock Exchange venture company that is working on steering the Canadian biopharma company in its first human clinical trials for chronic and subacute spinal cord injury patients. And we've heard just now from Dr. Jerry Silver, who was the inventor and now the scientific advisor to NervGen Pharma, about how we came up with this fascinating technology. And so I'd like to know how it's going to go forward. And, Mr. Mike Kelly, you've got quite an impressive resume from what I've been sent. You obviously, I would seem, have been hired to take this company forward. Tell me, if you could, a little bit about your background, how you came to know NervGen and where you're going to take it.
Sure, absolutely. So I've been in the pharmaceutical industry for roughly 30 years. I started out on the commercial side of the business in big pharma. And throughout those 30 years, I've broadened my experience from commercial. I've helped a lot of companies early in my career launch products, build out commercial infrastructures. Then I moved over into general management and got into the world of startups and biotechs. My last few jobs, I was at the C-suite level. One company, Covis Pharmaceuticals, we were a specialty pharmaceutical business that we ultimately ended up selling our products onto a Canadian company, Concordia, for a little over $1 billion. But my last job was with a company called Adapt Pharma, which was a very similar and a lot of analogies to where NervGen is today. When I joined, there was 12 people, and there's roughly that, a little bit more than that in NervGen today. We had a product called Narcan nasal spray that no one really heard about, and no one really cared about, except for the market. And we did a lot of hard work with a small group of people, ultimately developed that drug, launched that drug, and got millions of doses of Narcan onto the street in the US and Canada, and saved hundreds of thousands of lives over time. So it was a very gratifying role for me. Then post that, I actually did some consulting for a period of time. Was really kind of looking at life differently after you experience launching a product like Narcan. It's a life-changing product. You get very picky and choosy about what you want to do next. And really there was not a lot of things that were coming of mind and interesting. And then one day I was approached by a recruiter who said, you got to take a look at this company. They're based in Vancouver. And I scratched my head. I said, well, if they can regenerate nerves, why don't I, how come I haven't heard about them yet? And then I met Harold, and Harold was a fascinating story, was an entrepreneur. And I said, wow, this is a diamond in the rough. Harold then introduced me to Jerry, who you just heard talk, and I was sold. I'm not a scientist. I'm a businessman.
When was that, sir? How many years ago?
That was 7 months ago. I joined the company 7 months ago, and I think I talked to Jerry about 9 months ago.
Can I just interject a little story here?
Sure,
So,
go ahead.
it was a great call that we had, the three of us, and I think it went on for over an hour and a half. And I turned off the computer, the Zoom call, and I looked over at my wife and I said, "He's in. He's going to join us." And then Mike told me later he got off his Zoom call, turned to his wife, and said, "That's it. I'm going to join them."
That was it. That was exactly right. I did a lot of diligence, and I give the company a lot of credit for dealing with and being very patient with. That was my job.
Worth the wait. You were worth the wait.
So, congratulations for the decision for both of you. But I've had some experience in the pharmaceutical business. Raising money is pretty challenging. What's your strategy? You've got this phase 1, phase 2 clinical trial. You're hoping that you're going to get fast track and orphan drug status, but you might have to take it through phase 2 and phase 3. It's a long road. You've saying that Dr. Silver's already been working on this since the 1970s, the 1990s, depending on where you want to start ground zero from. How are you going to fund this?
Well, we've had, first off, let me, thank our current shareholder base. We have a group of investors who joined the company. The company went public very early on. Harold and Bill Radvac took the company public, I think a year after in-licensing the technology from Case Western, and a group of investors from Vancouver who have been very patient and very loyal, have really helped this company get to where it is today. And so I want to thank that group for all of their, continued support. But we've been growing the shareholder base. When I first joined the company 7 months ago, we were probably traded 80% in Canada, 20% in the US. And it's amazing how quickly, when a product gets into human clinical trials, how the word starts to spread. Right now, we're probably trading 60% of our shares are traded in the US and 40% are in Canada.
What's your market cap today?
Our market cap is just about 80 million US. I haven't looked lately, but we've gotten a little pop recently because we did announce that the study is underway. Several patients have been enrolled, and probably the biggest shot in the arm, which was a positive news for us, was last week when the FDA gave us fast track designation. So it's getting real. This is actually happening, and we're very excited. And what our strategy is just to continue to tell the story, to get as many people involved in the story as we possibly can. We're well funded today. We have a decent amount of cash. We have enough cash to get us through the readout of the chronic cohort.
So how many years of burn have you got?
We have just about a year of burn. Yeah. So we're the study that we're conducting right now. It's a small study, and we think post this data readout of the chronic cohort mid-year next year, money's not going to be a problem. Once we hit proof of concept.
So you've got these 40 people. You got these 40 people on the study in 2 different arms. And how long are they on drug? And you said you're going to read out next year. So how long have they been on the drug? And what do you expect to be the endpoint? That they can start walking again, or what? What's the endpoint?
Yeah, so all great questions. So the study is a 16-week study, and the patients are going to be treated for 12 weeks and then followed for another 4 weeks. So what we're trying to do is we're trying to mirror everything that we've conducted, or everything that was conducted in the animal studies, we're mimicking in the human studies. So the two cohorts, the chronic cohort and the subacute cohort, are identical to the way the data was done in the animal studies. We're doing a once daily subcutaneous injection. So this is injected into the belly fat, very similar to insulin. For anyone who's a diabetic, it's a simple once daily injection.
Do they do it themselves, or are they going to a clinical trial outfit?
Well, on the weekends, they're doing it themselves. During the week, they're going into Shirley Ryan Ability Labs, where it's a single center study done in Chicago. So Shirley Ryan is our partner. They're conducting the single center study. Monica Perez is our primary investigator. You asked about endpoints. So the endpoint, the design of this trial, the study was designed by Dan Michael, who's our chief scientific officer, who came to us from Amgen. Dan is a brilliant scientist, MD, PhD, and he's won awards with this study design. So the study design is to learn how this drug works and where this drug works. So the primary endpoint is actually called electrophysiology. So what we're measuring is electronically, we're measuring the signaling that's going from the brain to a hand muscle group and a leg muscle group. So how fast these signals are going from the brain to those muscle groups past the site of injury. So they're going to get a baseline, and then they're going to get treatment, and then we're going to measure them again at 12 weeks to see, will the strength of that signal increase? Will the speed of that signal increase? And if that does increase, the strength and the speed, that is an indicator that we are remaking connections. So that's the primary endpoint, is to remake connections. The secondary endpoints are all functional. You asked about walking. So we're going to have a walk test. We're going to have a 9-hole hand peg test. We're going to have grip speed, or pinched force. So there's a combination of primary endpoints which are electrophysiological, and then the secondary endpoints are functional. And our hope is to learn a lot in this clinical trial and make the connection between the electrophysiological and the functional, so that we could design the pivotal trial that you talked about earlier in the show.
And the pivotal trial would be when, how many people, how long would it take?
Well, it's going to be, it'll be a couple of hundred people, and it'll probably it'll take place as soon as we can get this data into the hands of the FDA and sit down and agree to an endpoint. So our goal is mid-year next year, this trial in the chronic cohort will have a readout. So we'll know what we're dealing with from a data perspective. It's going to take several months to get all of that data into a format that we can share with the FDA and get a end of phase 2 meeting with the FDA. And since now we have fast track designation, it means that we're going to have more open communications with the FDA, and we'll help, design a clinical trial, which is the next, the pivotal clinical trial, which should begin the following year.
Sometime in 2025.
Yeah, sometime in 2025 is.
And how long would that clinical trial be? Similar, half a year?
It depends on enrollment. If it's the chronic cohort, it's a pretty quick study. We expect, like this study that we're enrolling right now, we just announced enrollment in August. We'll be done enrollment in January, and we'll have a readout in June. So that's a pretty tight timeline for this study. The next study is going to be a multi-center study and more patients. So it may take a little bit longer than this current study, but it's, as far as timelines in central nervous system, this is as short as you can possibly get, because you have to think of it in the mechanism that Jerry talked about. We're measuring improvement. We're not measuring reduction in or degradation over time, which is a lot of studies in CNS measure. Well, we're measuring a difference over time of degradation. We're actually measuring improvement over time. So it's a shorter, smaller clinical trial. So
sounds absolutely fascinating, and obviously it's going to have a major impact if it's successful, and I hope it is. We're going to take a final break and come back with some concluding comments in just 2 minutes. Stay with us, everybody. Welcome back, everyone, to the Brian Crombie Radio Hour on Saga 960. Tonight, I'm introducing you to a really fascinating technology where Dr. Jerry Silver has developed a drug that is going to be injected into the body, ultimately maybe even an oral pill, but initially injected into the body. And it is going to potentially allow the regeneration of nerves in the spinal cord by, interestingly, taking away some of the body's naturally created blockages in the nervous system that strangely enough exist in the nervous system, don't exist in our arms and legs, our peripheral parts of our body, and don't exist in goldfish and some sort of other mammal in Australia that he mentioned that I can't remember right now. But it's absolutely fascinating. Dr. Panett, you told us really quite a heartbreaking story to start off about why you became interested in this in regards to your daughter-in-law. You know more about this technology probably than anyone other than Dr. Silver. You know about the clinical trial. You know about the risks. Would you have your daughter-in-law take this drug?
Absolutely. Interestingly, we haven't had that discussion yet. But I was thinking today that it's time that we do need to. So this will be the first, because she'll watch this, of course, in the next little while. And so, Cody, we're going to have that conversation. So I would recommend it for sure. I'm excited. I've seen, the science. Maybe we really haven't talked about the nature of the science in terms of its power, but Jerry's results are unsurpassed in the literature. Right? There's nothing really close in spinal cord injury, and he did talk briefly about these proteoglycans and their inhibitory effect. And just said quickly that it's in the brain and it's in the spinal cord. And what that means is that this technology has the power to do far more than just help people with spinal cord injury. And we do have publications in MS and stroke and other areas that are very robust. And so that's another exciting aspect of the company. But yeah, Cody, we're going to have that talk. So thank you, Brian.
CEO Mike Kelly, tell us if people want to check you out. What's the website they should go to? Do you have a presentation on that website about your company, and what stock exchange can they buy your stock at?
Sure. So our website is nervgen.com, nervgen.com. And we do have a corporate presentation. And as Harold mentioned, there's a lot of good information up there from a preclinical perspective. We have all of our spinal cord injury data up there. We have some fascinating videos that show you the preclinical animal models that are, astounding and eye-opening. We have some preclinical animal models in stroke that we'd love to get into next as well. So everything that you need to know about the company is up on our website. As far as trading our stock and investing in our stock, you can get to our stock two different ways. On the TSX, we are NV or NGEN.V. And we're also traded on the OTC in the US, NGENF. So our stock price is listed on both of those exchanges.
So do you have to file with both the OSC and the SEC?
Yes.
And Dr. Jerry Silver, I understand you're referred to as the Oracle.
Oh. Well, that was kind of a joke at an Asilomar conference in California. My former graduate student was chairing a session, a discussion about spinal cord injury and different strategies. And while she was presenting the history that preceded the meeting, that particular meeting, she put up a huge picture of my face and labeled me the Oracle. I was flattered to think that she thought of me that way, or that anybody else thinks of me that way. But a person who struggles as much as I did to convince the world that cartilaginous molecules that were inhibitory were present in the nervous system after injury, I guess it took something like an oracle.
Well, I bet you there's
just to persist all those years with those ideas.
I think Jerry's being a little humble here. Jerry is an oracle. You're going to hear a lot more about Jerry in the next couple of weeks and months as we progress as a company. Folks are going to want to know where this came from, and you'll see Jerry's name and face all over the internet in the next couple of months.
Well, I
Yeah.
tell you, there's a lot of people with spinal cord injuries and other injuries that are hoping your trial works out, that your drug works out, that it gets to market, and that Dr. Silver is seen as an oracle, and Dr. Punnett is seen as an incredibly smart Googler that found them through his internet searching. That's our show for tonight, everybody. Thanks for joining us. I really appreciate it. And, I think this is a really fascinating technology. And so I'm going to check it out. Hope you do too. Good night, everybody.